Hey doc, I think I've got an ear infection. Antibiotics are a wonderful medicine when dealing with bacterial infections, but unfortunately they're sometimes misused.
My car is making this funny sound. Chooka 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 chooka.
When antibiotics are misused, it can lead to them suddenly not working anymore. And if you ask folks like Richard Dawkins and Bill Nye the Science Guy, the answer is that this is incontrovertible proof of evolution right before our very eyes. It's an evolutionary arms race, they say. As our weapons of medical warfare get stronger and more sophisticated, so too the bugs we're fighting are getting stronger. They're evolving better defenses, making them immune to the best we can throw at them. The power of natural selection and random mutation laughs at our incompetent medical prowess. Bacterial evolution and antibiotic resistance is one of the all-time greatest hits regarding evidence for evolution. And it's true that antibiotics do sometimes stop working. And antibiotic resistance is a form of evolution, narrowly speaking. But is this a demonstration of the creative power of Darwinian evolution, or is there something else going on? Let's take a look more closely. Let's get to know these bacteria killers we affectionately call antibiotics. How do they keep us alive? Where do they come from? What's their favorite kind of ice cream? There's a lot of ways for antibiotics to defeat bacteria. Some explode it. Some gum up the works. Some starve the little bugs out. Others prevent them from multiplying, and so on. But there's essentially only one or two ways that bacteria can become resistant to antibiotics. The first and most common way bacteria gain resistance is that it's always been there. Antibiotic-resistant bacteria actually pre-exist the invention of modern antibiotics. There's an interesting story of some Arctic explorers from back in the 1800s. Turns out they froze to death before the discovery and use of antibiotics. Some morbid scientists found them recently and took samples of their gut bacteria and found that they already had resistance to some of our most powerful modern antibiotics. Just like some people are naturally short and immune to limbo bars, some bacteria naturally have a pre-existing resistance to antibiotics. Not because they've gained this ability after an encounter with the antibiotic, but it's just always been the case. That said, bacteria do have a neat special ability that short people don't have. They love to share. Not just cooking recipes and financial advice, but super personal things like their very own DNA. They'll swap loops of DNA called plasmids in a process called horizontal gene transfer, or HGT. Like third graders with Pokemon cards, they have no idea how they got them or even what they do, but they'll trade them back and forth, and occasionally, they'll share these resistance genes. Nothing new has been made or evolved though. Bacteria already had the ability to trade plasmids, and the genes for resistance already existed before antibiotics. This is hardly an arms race, and it's not a demonstration of Darwinian creative power, since there's no new mutations and no new genes that have been made. Not only that, but these extra resistance genes are frequently a net negative on fitness. Bacteria with extra useless genes tend to be less fit than regular old vanilla bugs. Imagine you're in a foot race. You usually don't need a duffel bag filled with camping supplies, a pickaxe, spare bowling balls, and a weed whacker during your typical foot race. If you do, great. Otherwise, it's a huge waste of energy lugging all of that stuff around, and it makes you less competitive. Bacteria with extra genetic load tend to be less fit than regular old vanilla bugs.
What about mutations though?
This is the second less common, but more interesting way that bacteria develop resistance to antibiotics. How does it work? Scientists are aware of pre-existing resistance. So to exclude it as a possibility in experiments, they can start with a single normal, non-resistant bacteria and let that multiply over and over again, and eventually the bacteria population can still develop resistance on its own. Here's how. When copying their DNA, organisms are pretty accurate and mutations are relatively rare, but given enough time, errors do sneak in, like typos or autocorrect when you're texting someone. Sometimes they're no big deal and you can get along in spite of them. But sometimes these mutations really do something and can confer resistance or even immunity to antibiotics. Amazing, that must demonstrate the mighty power of evolution. Well, hold your horses. It's sort of like someone who has no arms saying that they're immune to handcuffs. No legs? You're immune to toenail fungus. No teeth? Congratulations, you're immune to cavities. Think of how much you'll save on toothpaste and floss expenses. Deaf and blind? You're immune to getting annoying songs stuck in your head. Wow. This poor soul might technically be immune to those things in a perverse way, but it comes at a cost. He's hardly a more fit organism, and these mutated bacteria are just hobbled, busted up versions of the original. Darwinian evolution can't get very far by breaking things. Eventually, it'll run out of things to break. And in reality, when antibiotics are removed from the environment, these superbugs just sort of die out, because without the help of the antibiotics keeping the stronger, normal bacteria at bay, they can't compete and they die out. Those are the two ways that bacteria can resist antibiotics. Okay, forget about antibiotics. Let's look at the experiment that Darwinists always point to show evolution right before our very eyes. The granddaddy of bacterial lab work, Richard Lenski's long-term evolution experiment. In this famous experiment, Lenski has simulated in the lab, over the course of a few decades, more than 1 million years of evolution with bacteria. Since humans and other large organisms can take years or even decades to reproduce, it's difficult to see any change between generations happening in our lifetimes. But smaller microorganisms like the bacteria E. coli can reproduce in just minutes, letting us observe tens of thousands of generations and the changes that go along with that in real time. This is what Lenski has done for the past quarter of a century or so. Just let a bunch of E. coli bacteria reproduce and watched what happened. Pretty soon after the experiment began, he noticed that they started to grow much faster than normal. Then, a while later, Lenski's E. coli suddenly gained a new ability to eat something new called citrate. This was an earth-shattering find in the world of evolutionary biology. Scientists were swooning in the streets. To hear them tell the story, this was a huge new evolutionary leap, proving beyond a shadow of a doubt the power of random mutation and natural selection. But would you believe it if I told you there was a problem with this explanation as evidence for the creative power of evolution? No? Good, because that would be untrue. There are at least 3 problems with this explanation. What exactly happened to cause the E. coli to grow so much faster? Well, they broke their flagella. What is that, and why would it make them grow faster? The flagella is a little outboard motor of the bacteria. It helps them move around and do their thing. But Lenski's bugs were in flasks that were swished around constantly, allowing them to get nutrients without ever having to move. They were spoon-fed lazy bums. This usually vital part was not currently needed, so it was a waste of energy, and they got rid of it. It's like selling the wheels off your car when you're at a stoplight. You'll save a lot of gas that way, sure, but you haven't really made a better car, and it'll sort of be a problem the next time you need to go anywhere. Well, what about the citrate thing? It's gotta be obviously true that being able to eat a brand new substance is a good thing for these little fellas. Like if we could eat dirt or something. Imagine the possibilities. Well, to make a long story short, all of the complex genetic pathways and proteins needed to transport and metabolize citrate were there all along. Not only that, but E. coli also already had a sophisticated mechanism to turn this ability on and off depending on its environment, so that they could prioritize their optimum food source, glucose, and not waste energy. All that happened was that the sophisticated control mechanism, this switch, broke. This bacteria lost the ability to turn off an ability that they already had. Now it was stuck always on, wasting energy to try and transport and metabolize citrate even when it wasn't necessary. It's sort of like this alarm clock. It has the ability to make this lovely noise. It's a nice ability, but it can be turned on and off. But what if the alarm clock was broken so that it made that noise forever and could never be turned off? Much better, right? Right? How can this not be a win? Sometimes it's good for your alarm clock to make noise, but it's very good to be able to turn it off too. E. coli could already do a thing, and they were broken so that they did that thing constantly, whether they needed it or not. Maybe you call that evolution, but Darwinian evolution demands innumerable new genes, new proteins, new body plans, new organs, and complex characters that we see in nature. This is not that. It's equivocation, using language so ambiguously that it misleads. For example, Aristotle was Greek. Greek is a delicious kind of food. Aristotle must have been delicious. Evolution has so many definitions that it's sometimes difficult to pin down what is meant by it. One could mean a shift in pre-existing populations, like Darwin's finches or peppered moths. or breeding or cultivation like dogs or plants. But these are miles apart from extrapolating random mutation and natural selection can build new organs or body plans, or universal common ancestry, zucchini are related to tuna fish, which is even further apart from the wild claim that it all happens strictly through materialistic processes. Darwinists equivocate when evidence is presented to prove one definition of evolution, where there is evidence and everybody agrees, and then another definition is quietly swapped in based on wild extrapolations to hoodwink people who aren't paying close enough attention. To extrapolate from antibiotic resistance to the larger kinds of changes evolution would need to make new animals is kind of like saying, last week, this cow could jump 1 foot high. This week, she can jump 2 feet high. Doubling at that rate in just 30 more weeks, she'll be able to jump clear over the moon. It's a bad extrapolation. There's a hard limit to how high cows can jump. And there's a hard limit to the time and populations available to evolution, and to the number of features bacteria can break before running out of things to break. Darwin didn't write a book called How Pre-Existing Organisms Break Themselves. He purported to explain how novel species came about from a single common ancestor. And at least as far as this is concerned, it was a failure. Darwin can't get very far by squandering pre-existing genetic information. Darwin's mechanism creates variation largely by throwing genetic information overboard and selecting for damaged or broken genes, blissfully unaware that it's marooning itself on an island of genetic fragility, making organisms, ironically, less able to adapt in the future, thus hindering larger scale changes from accumulating. There's an old joke that perfectly summarizes what's going on. Wow, you're so rich. What's your secret to becoming a millionaire? Oh, it's easy. First start with $1 billion and then light a bunch of it on fire. Bacterial evolution is lauded as a premier example of Darwinism in action, as Dawkins puts it, before our very eyes. And actually, it is. It doesn't demonstrate Darwinism's creative power though. Instead, it's a perfect demonstration of Darwinism's impotence. Alright, imagine we're a long time ago and life isn't around yet. We've got some of the building blocks needed for life just hanging out. What are the next steps in chemical evolution that we would need to turn these little dudes into something actually living? Forming biopolymers. What's a biopolymer? That's when our building blocks team up and link together in long chains. This is what life's molecules are made out of, from DNA and RNA to proteins. Everything needs them to live, so biopolymers are kind of important. In order for chemical evolution to have taken place, nature would need a way to assemble biopolymers. But like hiding your tuna sandwich in a room full of cats, it's a very hard thing to do. Some scientists claim that it was no big deal though. They contend that under the right conditions, making biopolymers is easy enough, and from there, life could flow naturally. But is this really the case? Let's take a look more closely. These building blocks are kind of like fish. They don't have legs. And without something to float around in, they can't get around like they need to in order to do stuff. Because all known life requires water, it is commonly seen as the solution to this problem. Water allows these molecules to bump around and knock into each other, which could help link the monomers into a chain. So, water is needed, but water is actually a big problem as well for 2 reasons. Like bringing your mom with you on a date, water can slow down or stop chemistry. Water molecules dilute the building blocks, pushing them further apart from one another, making it less likely that they'll be able to interact. Also, water actively breaks down these polymers. Bazillions of water molecules are speeding around and constantly smacking polymer chains around. This frequently breaks the bonds, cutting the chain to pieces, something called hydrolysis. We need a liquid for these building blocks to link up, but the liquid also prevents and destroys the links. That's what makes it a paradox. Some have suggested that wet and dry cycles, or salts, may be able to fix some of these problems. However, wet-dry cycles actually degrade DNA or RNA by removing nucleobases, creating abasic sites. Formation of a biopolymer chain is actually very difficult in wet-dry conditions. Proteins in particular have an additional challenge. Like a balloon animal that relies on air to support its shape, proteins rely on water to support their complex three-dimensional form. When you remove water from a protein, it tends to fall apart, something called denaturation. Many proteins will just irreversibly collapse when dried out. Well, if water is such a problem, what if we just used a different solvent? Geochemists laugh at the idea of large amounts of a different solvent on Earth. We just don't have pools of formamide or methane or whatever lying around. Also, all of the other potential solvents tend to be very volatile as well. They evaporate quickly, or they're only produced in low concentrations, or they're only liquid at temperatures that are incredibly hostile to life, or they're nonpolar, or they're typically produced by biological activity. In other words, there are a lot of problems with using something other than water, and you can't get many of these solvents needed for life unless you already have life. More complex molecules frequently have something called chirality. You can have the same molecule just in different forms, like you have two hands, one left hand and one right hand. All but one of the 20 amino acid building blocks have chirality. Except for glycine, he's pretty silly. The crazy thing about life is that all proteins, all DNA, all RNA, all cell membranes must have the correct chirality or else life would cease to exist. Think of it like molecules having an evil twin. Here is a portion of insulin. According to this paper, if any one of these had the evil twin, the wrong chiral form, it could no longer form the 3D structure properly. If you have a long molecule like DNA, you don't just need the right chirality once. It has to be correct in every one of the 500,000 nucleotides in the simplest free-living form of life. Well, what about these papers? They show ways to preferentially filter or select one chiral form over another. So I guess it's not that big of a deal. Actually, the most successful proof of concept is from this paper, but it's an open secret in the field that the chemistry that's required for this method couldn't have happened on a prebiotic Earth.
A lot of people, our group included, are trying to find more prebiotically relevant reactions that could do what the soy reaction does. So far, we haven't been able to find one, so that's a kind of a holy grail.
This experiment and others like it are interesting, but basically classified as prebiotically irrelevant. Natural processes are well known to produce equal portions of the two different chiral forms. That's what nature does. Filtering out only one form over another is very unnatural. There's no process that we know of on the prebiotic Earth that could have done this. But it gets worse. It's not as simple as having an evil twin, just left or right-handed chirality. Adenosine, for instance, has 15 evil twins, or 16 chiral forms. It's the identical chemical formula to the other nucleosides. It's still C10H13N5O4, but if you slot one of these evil twins into a creature, bad stuff happens. Fun fact. Platypus venom contains a protein that can flip amino acids of its victims to the wrong chirality, causing devastating pain. You dirty little chirality flipping platypus. Every biopolymer has a chirality problem. So how does modern science deal with it? Well, they largely ignore it. You could read thousands of pages in college-level textbooks or popular works, specifically on biology and the origins of life, and never even come across the concept of chirality. Diagrams that we find in our textbooks may make it seem like there's only one way for things to link up, but linking together different building blocks is hard. The reality is that there are far more ways for these little dudes to link together incorrectly than correctly. Specifically, there's 4 different kinds of ways that it can go wrong. Number 1, intramolecular cyclization. If the bit at the end links to the bit at the front, making a circle, it's lights out for that polymer. He's dead. Number 2, heterogeneous impurities. Interfering molecules or the wrong molecule could again spell death for a fledgling polymer. 3, branching malformation. You could have the right thing but have it linking to the wrong spot on another building block. Or number 4, premature truncation. There are monomers that act as terminators. Not that kind of terminator. They prevent further growth. Polymers can go wrong in so many different ways, and the longer one gets, the probability of achieving correct links goes down exponentially. Anyway, with all of the smart people working on this problem for so many years, how's the progress coming? Let's take a trek through science.
Trek through science!
Well, in the 90s, they were able to make a chain, a polymer, that was 11 monomers long. More recent experiments showed that they were able to get a chain up to 50 monomers long. Progress. Unfortunately, more recent papers have shown that these claims were wrong and not reproducible. But regardless of whether they've made a polymer that is this long or this long, even that is with extensive coddling and unnatural cheating. See this video for more information about that. When we remember that the simplest known free-living organism's genome is more than 500,000 nucleotides long, the argument sort of becomes moot. Life is so far in the distance as to be imperceptible. If, by some miracle, a polymer was able to contend with the water paradox, the chirality problem, and linkage issues, it's got a whole lot more to worry about. It has to face deadly heat, UV rays, mechanical wear and tear, ionizing radiation from the Earth, free radicals, and other forces waiting to pounce and break things down. But if you had enough time, though, surely anything could happen, right? Meet Manfred Eigen. He realized that the information in RNA or DNA that's required to maintain life would experience an error catastrophe without complex enzymes that can recognize and fix this damage to DNA. Without these error correction systems, you would essentially die of old age before you were even born. Fortunately for us, all living things are equipped with complex repair mechanisms. Unfortunately for abiogenesis, these mechanisms need to be encoded within DNA, but the DNA can't exist without the repair mechanisms. This is Eigen's paradox, a chicken and egg problem that has been around without a solution for decades. We can't have long strands of DNA or RNA unless it can repair itself, but it can't repair itself unless you have long strands of DNA. Producing any biopolymer through purely natural processes is a huge, frustrating, intractable challenge to science. But life is not based on a few random biopolymers, though. Life requires thousands and thousands of highly specific biopolymers with very precise arrangements of monomers. Even if you could get biopolymers to form by natural processes, keeping them intact is quite another challenge. RNA is so unstable that it needs to be kept in deep freeze, away from sunlight, and it has to be thrown out if it's left at room temperature for more than just a few hours. RNA spoils about as fast as a tall glass of milk on a hot summer day. And we're supposed to believe that molecules like RNA not only existed for millions of years, but also replicated and became more complex. You can believe that if you want, but it'll be in spite of the evidence, and not because of it. Charles Darwin and some modern scientists think that life started all on its own. It sort of just happened spontaneously. It's called chemical evolution. They say it started with chemicals forming building blocks and then combining into polymers like DNA and RNA. These previous videos detail just some of the scientific problems with that ever happening. Anyway, let's wave our little magic wand and say all those challenges have been solved for now. We don't have life yet, but what are the next steps toward life? Well, just like eggs have a hard time turning into a chicken without a shell, and water balloons are not much fun without the balloon, living things need a cozy little house of their own to keep the good stuff in and the bad stuff out. We're talking cell membranes. All living things are made up of cells that are surrounded by a cell membrane. Life simply cannot exist without membranes. Origin of life researchers claim that cell membranes are easy to make and that they have demonstrated this in the lab. They point toward experiments where these molecules called phospholipids are placed in water and they form into a spherical shell, kind of like a cell membrane, all on their own. Wow. But is it really the case that cell membranes are so easy to form? Let's take a look more closely. Real quick, if you're new here, Long Story Short is about explaining science in an easily digestible way. If that sounds good to you, consider sharing this video with a friend and subscribing for more. Okay, cell membranes, needed for life, supposed to be easy to make. Is that the case? Membranes do a lot of essential things to keep the cell alive. If any are missing, death results. Think of the membrane like a house in outer space. Space house, nice. Space house provides a safe, comfortable environment inside, despite things outside desperately wanting to kill you. Outside, the temperature can fluctuate between as hot as an oven and as cold as negative 455 degrees. You know, almost as cold as your ex's heart. Not to mention the radiation, lack of oxygen or water, there's a vacuum, space pirates. Inside though, there's an ample supply of pizza, tacos, warm blankies. That's what I'm talking about. The environment inside the house is controlled and very distinct from the outside. That's what keeps us alive. In a cell, the membrane is responsible for keeping things safe and comfy too. The fancy name for this is homeostasis. But walls that are sealed up tight with no way to let trash out or new food in will quickly become a stinky tomb. The cell will quickly die as food runs out and garbage piles up. The very simple membranes that scientists create in the lab are exactly like this. Non-functional, stinky tombs. Real cell membranes need to be tight, but also have very specific doors that actively let things in and out in order to maintain homeostasis. And it needs to have all of this complexity at the very beginning. There's no time for this to gradually evolve because no homeostasis, no life. What if early membranes were a bit leaky, like these papers suggest? Maybe the first cells had holes in them that were just big enough to let good things in and keep the bad stuff out, somehow finding a good balance to maintain homeostasis. Unfortunately, that won't work. All existing cell membranes need to let big things in, while at the same time keeping small things out. In other words, they have conflicting requirements. All cells harness their energy by pumping protons out and very carefully letting them back in to spin these proton turbines, like a hydroelectric power plant. And protons are teensy tiny. To give you a sense of scale, if a proton were the size of a housefly, then a bit that the cell would need to let in, like this amino acid histidine, would be roughly the size of the country of Monaco. And the smallest known cells would be roughly Sweden-sized. Those are the approximate scales that we're working with. Basically, Sweden would need to keep houseflies out, while at the same time letting the Monaco-sized things in. If the membrane let too many fly-sized protons slip in, the cell would die. So the membrane has to be tight enough to not let these teensy-weensy protons in, but also porous enough to be able to let these relatively gigantic things come right on in. These conflicting requirements of the membrane are a real head-scratcher. Fun fact, at any given time, a cell is generating roughly the same voltage per meter across its membrane as a bolt of lightning. Another conflicting requirement is that cells have to let water in, but if water was just allowed to pour in willy-nilly, so would those sneaky tiny little protons, and this would drain the cell's battery, so to speak, and the cell would, again, die. Thankfully, cells have specialized doors called aquaporins. They work sort of like an airlock in our space house by safely letting things in without destroying the nice environment inside. The aquaporin lets water molecules in single file, but that still wouldn't be enough to stop the protons from sneaking in and killing the cell, because the water acts sort of like a wire, conducting the protons along itself. So the aquaporin also rotates each water molecule by 90 degrees, flipping them around and eliminating its ability to conduct protons for just long enough to safely let the water inside. Every cell has tons of these incredibly specialized doors. The simplest known free-living life form is a single-celled guy called Mycoplasma genitalium, but it has thousands of specialized doors composed of over 100 different proteins that operate within the cell membrane. So even the simplest cell has an extraordinarily complex membrane, an active, selectively permeable boundary that allows and often forces exactly the right things in and out to maintain homeostasis. A perfect environment inside the cell, despite wildly varying conditions outside. leaky membranes and tight membranes would both kill the cell. The membrane had to be extremely complex from the very beginning, or life could never begin. Well, yeah, today it's very complex, but there's been a lot of time for things to evolve. When life was just getting started, there weren't cells, there were proto-cells. They were way simpler. Okay, could life have been simpler? Scientists try to gauge what the simplest possible life form could have been by taking a very simple cell and removing as many things as possible without killing it. These papers took a very simple bacterial cell that had about 985 genes and cut it down to 474 genes. But the bug was barely hanging on, so they added a few genes back in to get a cell that was more reasonably alive. They found 493 genes were essential to keep the minimal cell alive. This work suggests that there is a practical boundary for how simple life can possibly be, not just today, but at any point in time. But in order to cut that many genes from the original 985, they essentially had to coddle the poor, hobbled little bacteria. You see, the original bacteria was able to make its own food from what was around it, like a big boy. But the downsized bacteria needed someone else to make his PB&J for him, and he was picky. He needed it cut into triangles and had the crust cut off. Dad, how many times do I have to tell? It required a precise cocktail of nutrients, basically life support, or else it would die. However, most of these life support nutrients couldn't have existed on a prebiotic Earth. If you try to simplify a living organism, removing some of its tools for survival, it doesn't just magically no longer need that thing. The only way to keep it alive is to place a heavier burden for survival on the surrounding environment. The organism just becomes more genetically fragile, unable to endure slight environmental changes, or it'll become dependent on other life forms to keep it alive, which, if you're paying attention, is not a possibility for the first living thing. Consider yourself. Could you live without legs? Technically, yes. But more requirements will be placed on your environment. You will need a wheelchair, ramps to go up or down, and nice smooth surfaces. Or robot leg. Can you live without kidneys? Technically, yes. But you will need to be hooked up to a sophisticated dialysis machine to do what your kidneys do. 3 times a week for 3 to 5 hours per day for the rest of your life. The more you remove from a person to simplify them, the more requirements are placed on the environment to keep them alive. The same is true for microscopic life. These experiments don't actually do away with needed complexity, it's just exported to the environment. Real cell membranes are complex machines with many required functions, like identifying needed molecules and opening specific doors to let them in, or force them in, identifying waste and exporting it, maintaining proper pH, salinity, and osmotic pressure, blocking toxins and dangerous invaders, providing electrical insulation, energy harnessing, and even acting as a template for expanding the membrane during replication, because cells can't actually build a membrane from scratch. They need a pre-existing membrane to do that. The lab-made membranes, on the other hand, do literally none of these things. They're closer to dumb soap bubbles or salad dressing than anything else. When scientific literature or textbooks claim that membranes are easy to create in the lab, it's a lot like saying the Mona Lisa is easy to draw. Or making some dinosaur-shaped chicken nuggets is equivalent to making real actual dinosaurs. There's a bit of a difference there. The observable science makes it clear that, 1, because of the need for homeostasis and the conflicting requirements of cells, the membrane had to be extremely complex right from the very beginning, or life could never have begun. Claims that this problem is even close to being solved are badly misrepresenting the science. And 2, attempting to simplify a living organism merely kicks the can down the road. These experiments only hide the complexity by exporting it to the environment. And a prebiotic environment was certainly not like a lab or hospital ICU. So, when you hear protocell, think leprechaun or rainbow unicorn. There is no evidence that any of these ever existed. And the more science learns about life, the more unscientific chemical evolution becomes. Could life have started from simply natural processes? Little bit of chemistry here, dash of physics there, a lot of time, and boom, we've got the beginning of life, abiogenesis, right? We've already detailed a number of serious issues with abiogenesis in these videos. But just like your favorite superhero movie, let's briefly ignore the laws of physics. We'll just assume that all of the scientific challenges mentioned in those prior videos have been vaporized. So, we now have lots of building blocks, biopolymers, and a cell membrane that is fully equipped to maintain homeostasis. Awesome. We still wouldn't have life yet, because life is built around something called gradients. A gradient is basically more of something over here and less of it over there. The opposite of a gradient is equilibrium. Physics loves equilibriums, but not so much gradients. For instance, water likes to be in equilibrium, all spread out evenly. But if you build a dam, you can create a gradient, collecting a bunch of water on one side and not a lot of water on the other side. The water naturally wants to flow down from the high side to the low side, and this is where energy can be harnessed. Life does the same thing, except instead of water, cells use proton gradients to capture energy. It's super important. No gradients, no life. To advance further toward life, we would need a continuous supply of these energy gradients and a way to harness them. Some scientists claim that there were plenty of energy sources to support the first proto-life forms. Hydrothermal vents, the sun, lightning, hydrothermal vents, volcanoes, hydrothermal. Is it really the case that life could have started with these kinds of energy sources? Let's take a closer look. It's true, the universe has plenty of raw energy out there, but there's a very important difference between that and what life can actually use to exist. Life is pretty picky. It needs very specific kinds of energy, and it needs to be fed it in a very particular kind of way. Otherwise, it's kind of like trying to charge your cell phone with a charcoal grill. There's lots of energy in the grill, but none of it is usable for your poor, dying phone. To harness the raw energy of the grill into something your phone can use, you would need a set of pretty complex machines. A boiler to heat water into steam, a turbine that the steam pressure can spin, an electric generator, then transformers, converters, regulators to get the energy in just the right form. And then you need the right kind of charger plug thing for your phone, and if you're lucky, the cable's long enough so that you can lay in bed while it charges, and that's a weird angle. But, like life, your cell phone isn't just picky, it's also fragile. Not only will the grill not charge your cell phone, but it'll also destroy it. If the little circuits and bits in your cell phone get too much energy, or even a little bit of the wrong kind of energy, it's done for. Ooh, battery's almost dead, let's get you some energy. Energy! 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 Raw forms of energy can only be used for life if complex machines are available to harness them, and the wrong kind of energy will destroy life. Okay, maybe life didn't get started in a volcano, how about just nearby? Hydrothermal vents are a perfect example. They're a great source of constant energy gradients that early life could have used. Hydrothermal vents aren't like lightning, they're more of a gentle, sustained energy source. Yeah, that is true, but living things can't just absorb energy like a sponge, even if it is gentle. Even the simplest forms of life harness energy through a 3-step process. The fancy term for this process is romantically known as chiasmatic coupling. First, 3 proton-pumping complexes known by their friends as NADH ubiquinone oxyreductase, coenzyme Q cytochrome C oxyreductase, and cytochrome C oxidase. These guys are also known as the respiratory complexes, and together are made of about 25 different proteins, each of which is very fancy in its own regard, consisting of hundreds of precise amino acids. Anyway, these proton complexes use electrons that are stripped off the cell's food source to power machines that pump protons, known as proton pumps, resulting in fewer protons inside the membrane and more protons outside, which conveniently provides an electrical voltage, or a gradient. The respiratory complexes have to be complex because stripping electrons is a dangerous process, like handling a bomb. If any of the 15 reaction steps in this process are mishandled, the loose electrons will destroy the first molecule they encounter. But physics is pretty unhappy about these protons getting kicked out of the cell membrane club and wants to shove them back in. So, step 2. The disgraced protons are only allowed back into the cell if they first turn an electric generator called ATP synthase. In the simplest form of life, ATP synthase is a machine made from at least 20 interconnecting proteins, and it requires other proteins to help assemble it too. complexity upon complexity. Nick Lane, an Origin of Life researcher and biochemist at University College London, describes ATP synthase as the most impressive nanomachine of them all. It is hard to convey the astonishing complexity of this protein motor. This is precision engineering of the highest order, a magical device, and the more we learn about it, the more marvelous it becomes. Rotation of this ATP synthase motor is used to recharge dead batteries, ADP, turning them into ATP. It doesn't look all that complicated until you realize it looks more like this. And this is slowed down by more than 10-fold. And the cell membrane is so densely packed with these machines, they effectively make the entire surface of the membrane. Step 3. The charged up ATP is plugged into a wide variety of other biological machines to power all the living stuff we need to do, like building proteins, repairing DNA, replication, cell division, blinking, subscribing to this YouTube channel, you name it. When the ATP is discharged and turned into ADP, it gets sent back to the ATP synthase to get charged up again. Fun fact, a typical human generates and uses roughly 1 billion trillion ATP molecules per second. That's your entire body weight in ATP every single day. Whoa. Here's the paradox. The cell needs to make ADP, the empty batteries of life, before they can be charged up to ATP and put to use. But producing one new empty battery requires at least 7 already charged batteries. So you can't make ADP until you already have ATP around in the cell. And ATP is required to build ATP synthase. But ATP synthase is required to charge ADP into ATP. So life's energy harnessing process is one big paradox. You need it before you can have it, and you can't make it until you've already made it. Every part had to arrive, assemble, and start working all at once, leaving no time for it to gradually develop. Hang on a second. Hydrothermal vents are supposed to solve some of these problems, though. There are 2 types of hydrothermal vents. The first are called black smokers, and they do provide raw energy and some very simple molecules that early life would have needed. But on the other hand, black smokers are too hot and too toxic. Any organic compounds that form at these temperatures would quickly degrade back to CO2. They're also very unstable, growing and collapsing over a few decades at most. Not nearly enough time for anything useful to happen, even if it could. They're not seriously seen as a solution to the problem of chemical evolution. The other kind of hydrothermal vents are called white smokers. These are instead proposed to be the solution. They're not as hot, and they're better suited for harnessing energy because of a free pH gradient. And the rock structure provides tiny membrane-like compartments for fledgling life to take root. There are a few big problems with white smokers as well, though. A simple cell requires 100 million times the energy density that could be produced by these alkaline vents. And as you probably guess, rocks make poor membranes. The rocky membranes in alkaline vents are hundreds of times thicker than the membranes of life. The pH gradient across such a thick membrane would not provide enough useful energy for life. So rather than protecting and nurturing life, these white smokers would strangle it out instead. The high pH also removes the carbon that life needs and is destructive to RNA, and on top of all of that, we would still need complex machines to harness the energy. White smokers do nothing to solve that problem, and scientists are well aware of this difficulty. But life started in a much simpler form. It didn't have to start with something so complex like chemiosmotic coupling. Just look at fermenters or methanogens and acetogens. For harnessing energy in life, fermentation is the only known alternative to chemiosmotic coupling. It produces a small amount of ATP through a simpler process called substrate-level phosphorylation. But this kind of fermentation is not sustainable. You see, strict fermenters pollute their environment, which quickly stops them from growing or reproducing, unless their waste is cleaned up by other forms of life that use chemiosmotic coupling. So this effort to simplify only increases the total complexity. Methanogens and acetogens don't use respiratory complexes to produce their proton gradients, but they still require complex enzymes to produce a proton gradient, and they still rely on the incredibly complex ATP synthase to generate their ATP. So again, this doesn't really simplify the problem. Okay, what if earlier life didn't use ATP? Then it would just need a different set of complex machines to harness whatever other energy it used. Then you would have to explain how those machines transformed into the machines that all of life uses today. In case it needs to be said, there's no evidence for this, but more importantly, it actually makes the problem worse. But we've got lots of time. Time won't help you because you can't store ATP. These things are so unstable, particularly around hydrothermal vents, it'll basically decay instantly. And it's hard to overstate how much ATP is needed every second of every day in order to sustain life. Even if aliens dumped off a big tanker of ATP, it wouldn't help if we didn't have the machines to plug it into, or a way to recharge it. Life requires a continuous supply of energy that must be converted into just the right form by a series of complex molecular machines. And the complex molecular machines themselves could only have been produced by an established energy harnessing process. This is yet another paradox for the origin of life. Imagining that life started simpler and had lots of time to accomplish this is a half-baked idea, like trying to charge your cell phone on a charcoal grill.